Hello students, how are you all? Are you following the classes? Okay. From this session, classes will be on digital mode. So let's start the class. Okay. So in the previous classes, we have learned about the cryptogams. So cryptogams are Thallophyta, Bryophyta and Pteridophyta. In Thallophyta, no plant-like organization is seen and they do not have any vascular bundles. Okay. No. Now in Bryophyta, they have plant-like organization, but they are phylloids, colloids, and rhizoids, and they do not have any vascular bundles. Okay. Next, coming to the pteridophyta, they have true roots, leaves, and stem. They have also the vascular bundles. So these are called as vascular cryptogams because these are the first true land plants with leaves, roots, and stem, and also the vascular bundles. Okay. So you can see the ascending pattern of characteristics or the evolutionary characteristics which are developing from Thallophyta to Pteridophyta. In next, in the next division, okay. So in the next division, that is in phanerogams, we have gymnosperms and angiosperms and angiosperms. Both of these are well developed. Both of these have well developed plants and they also have well developed vascular bundles and these show secondary growth. Okay, secondary growth. Now coming to the gymnosperms, we have already talked about the morphological characteristics of pinus in the last class. Okay, so we have discussed all the leaves, stem and roots, but we didn't discuss about the vascular bundles. So the gymnosperms have vascular bundles of collateral, conjoint, and open type and open type. Conjoint means xylem and phloem together in a single vascular bundle. Okay. Next, coming to the collateral, this means xylem and phloem both on the same radii. Okay. Next, open. What is open? If there is a cambium or cambial wall between the xylem and phloem, so this type is called as open type. If there is no cambial wall, it is called as closed type of vascular bundle. Okay. So these are the some of the ascending patterns or sorry, these are the some of the ascending pattern of developmental characteristics you can see from Thallophyta to gymnosperms. Okay. Next, coming to the root system. In gymnosperms, root system in gymnosperms, the root system is associated with microorganisms. Okay. So in cycas, we can see coralloid roots. So these coralloid roots are associated with blue-green algae. So these blue-green algae, they help in the fixation of nitrogen. That means it can fix the nitrogen for N2 fixation. And in return, these algae, that means these blue-green algae, they get food and protection from the plant. Food and protection. Okay. Next. In pinus, roots are associated with mycorrhizae. Mycorrhizae. These are a special type of fungi, you can say. So these help in phosphorus assimilation or phosphorus fixation to the plant. It can fix phosphorus to the plant. Okay, in return it gets protection and food from the plant. Okay, so these are the special type of characteristics we can see in the roots of gymnosperms in cycas and pinus. Okay, next coming to the male cone. Male cone of pinus. We can say it as a strobili or strobilus. Okay, strobilus is the singular and strobila is the plural. Okay. So how this male cone is formed? This male cone is formed at the tip of the plant, tip of the branches, you can say. So it's not the exact tip, but the just below the apical bud. Just below the apical bud. Okay. So these are formed in bunches. These are formed in, that means the male cone, sorry, male cone is a bunch of leaves or it is made up of leaves, and these leaves are present at the tip of the long shoot and in clusters. Okay, so these are formed in clusters. Okay, so these leaves, male cone is formed of a cluster of leaves. So these leaves are called as microsporophylls. Microsporophylls. And these microsporophylls, they produce microspores. Okay. So let's see the male cone. So these are the leaf-like structures which are present on the male cone. Okay. So these are the hard, woody leaves, and this is the axis. Okay. Let's see this is the male cone. Look, it is the cluster of hard woody leaves. Okay. Let's see the LS of male. Okay. So these are the microsporophylls. Microsporophylls. And you can see the microsporangium inside it. Okay. The blue circles are microsporangium. These are the microsporangia or microsporangium. And this is the axis on which these leaves are arranged. Okay. So this is the LS of male cone. LS of male cone. Okay, now let's take a single microsporophyll. Okay. So this is the hard woody structure or microsporophyll. Microsporophyll. This is the sporangium. Sporangium. And these are microspore mother cells. Microspore mother cells. Microspore mother cells. Okay. So these microspore mother cells, let's say MMC, microspore mother cells, they produce microspores. Produce microspores. Microspores. And then these microspores, they change to pollen grains. Pollen grains. Okay. So these pollen grains look somewhat like this. Okay. This is the outer exine. This is outer exine. And this one is the intine. This one is the intine. That means the outer exine or outer layer. And this is the intine or inner layer. Okay. These winged structures help in pollination. Okay. So these wings, 1 and 2, these are the wings. So this is the wing-like structure. They help in pollination. Okay. Help in wing-like structures. Wing-like structures. They help in pollination. Okay. Next, through wind. Through wind. Pollination is carried out through wind. And this pollination is called as anemophily. Anemophily. Or anemophilus. Anemophilus. Okay. Next. Sometimes these pollen They germinate and produce the pollen tube in the sporangium. These pollen, they produce germinal tube, or they germinate. They produce pollen tube. That means they germinate in the sporangium. Sometimes in the sporangium. Okay. Large number of pollens are produced. Why? Because large number of pollen are produced because if wind blows in wrong direction, then there should be some extra pollen for the pollination. If wind is blown in wrong direction, then the pollen are carried away by wind. So there would be no pollen for the pollination. That for that reason, these pollen are produced in large numbers. Okay, so large number of pollen grains are produced. Okay, so color of these pollen grains are sulfur yellow. Sulfur yellow. Okay. Next. At the pine fields, there would be the sulfur clouds. At the pine fields, there would be sulfur clouds. That means the yellow clouds are seen at the pine fields. Sulfur clouds. Okay. So these are formed by the large number of pollen production. Okay. So this is the male cone. This is about the male cone. Now let's take a look of female cone. Female cone. Or you can say it as strobili. Or strobili. Okay. And here you can see megasporogenesis. Megasporogenesis. Okay. Here larger, these are, okay, the female cones are larger than male cones. Larger than male cones. And they grow, and these grow in the axil of, these grow in the axil of the scale leaves. Axil of scale leaves. In the previous class, we have already discussed about the scale leaves and the folia leaves. Okay, scale leaves are present on the stem all over the body. Okay, so in the axils of these scale leaves, these female cones are produced. Okay, they grow in clusters. These also grow in clusters. These grow in clusters. That means how many female cones can we see is 80 to 90 megasporophylls on axis. We can see 80 to 90 megasporophylls. Megasporophylls on the, okay. Development of this female cone can take up to 3 years. Okay, that means the female cone develops from 1 to, it takes time from 1 to 3 years. Why? Because there would be a difference in the cones of first, second and third year. We can observe in the leaf gaps around the axis. You can see so this is the axis and these are the first year of female cone. Here there are no gaps. Okay, no gaps. First year of female cone. 1-year-old female cone, you can say. No gaps in between these megasporophylls. Okay. In the second year, you can observe there is a gap. That means the megasporophylls are growing. There would be some gaps between them for the growth. Okay. In the third year, this is the axis, and these are megasporophylls. There would be lot of gaps between these megasporophylls. The third year, you can see this in the third year. 3-year-old female cone. Okay. Now, these cones take very long period to develop. That is from 1 to 3 years, as we already said. Okay. Now, you can observe the gaps between these megasporophylls, so that you can say whether it is a 1-year-old, 2-year-old, or 3-year-old female cone. Okay. So now enlarging the megasporophyll. Let's enlarge the megasporophyll. Enlarge one megasporophyll. This is A. This is the larger woody structure. This one is the large woody structure. And this one is the bract scale. This is the bract scale. Okay. So this is ovuliferous bract. This is the ovuliferous bract. Okay. Now, this bract scale is for holding the pollen. Holding the pollen. At last, pollination occurs in the female cones. So, there should be some structure to hold the pollen for the pollination. Okay. Next. The time period. Okay. So, the time period for pollination is, it can take 15 months. Sorry. The time period between pollination, this is between pollination and fertilization. Pollination and fertilization. That means a pollen grain has reached here, and you can see here the ovules are present. So these ovules produce eggs, and the pollen fuses with the egg. So this fertilization step will take 15 months of time period from the pollination. Okay. So pollination and fertilization takes 15 months. Okay. Let's draw the so this is the megasporophyll, and here you can see the pollen. Okay, which are stored in the bract scale. These are the pollen, and you can see the ovule in the ovuliferous bract. So this is the ovuliferous bract, and here you can see the ovule, and these are the integuments, and this is the okay. And these pollen grains, at the time of fertilization, they germinate, they produce pollen tube, and they reach ovule for the fertilization. Okay, let's enlarge one of the megasporophyll. You can see here the ovules. So each megasporophyll have one or two or many ovules. Okay, so these are the integuments, and you can see the ovule. And in gymnosperms, you can see unitegmic ovules. In angiosperms, you can have bitegmic ovules. Okay, so you can see here unitegmic ovules. And these are orthotropous unitegmic. That means it has only one integument rounded. Okay, and this has a single egg, and this ovule has a single egg, which is the female gamete. Okay, so this is about male and female cones of pinus. Okay, so in the gymnosperms, you can see another example is cycas. Let's see the overview of cycas. Let's see the overview of cycas. This is the cycas. And they grow in warm climatic conditions. They grow in warm climatic conditions. Grow in warm climatic conditions. And the stem is called as cordex. It is unbranched structure. Unbranched structure. Okay. So leaves are present at the tip. Leaves are present at the tip. And the arrangement of these leaves are called as crown. Okay. And these leaves are foliar leaves. They are green in color, and they can perform photosynthesis. They can perform photosynthesis, and these leaves are pinnately compound. Pinnately compound. And this cycas plant looks somewhat like this. Is like a coconut tree, and these leaves are these. These leaves are called as pinna, and one single leaf is called as pinna, and this complete leaf structure is called as compound leaf. Compound leaf. So this leaf is called as, effectively together it is called as pinnately compound leaf. Innately compound. Okay. You can see like this. So the arrangement of these leaves on the tip is called as crown. Okay. So these scaly leaves, these are present on the stem. These are present on the stem, and these are brown hard structures which covers all over the stem. These are brown hard structures. Hard structures. So you can see them here. So like this. These are the brown hard structures called as scale leaves. And these are present all over the stem. Okay. Now coming to the reproductive organs. And these plants are dioecious. These plants are dioecious. That means it has male and female plants separate. Okay, separate plants which has cones by males are called as microsporophylls. Microsporophylls. And females have megasporophylls. Megasporophylls. In cycas, the females have no cones. So no cone formation is seen in females. Okay. So these microsporophylls are formed on male tree, and these megasporophylls are formed on female tree. Female tree. Okay. Next, coming to the next point. In cycas, we find the largest male gamete and the largest female gamete and the largest ovule. Okay. These are the largest male, female gametes and ovule of the all over the plant kingdom. These are the largest of all the plant kingdom. Okay. So what you can find in this? You can find a largest male gamete and largest female gamete and the ovule. Okay, so these are found in gymnosperms. Found in gymnosperms, that too in cycas. So these are the largest in whole plant kingdom. Okay, whole plant kingdom. In gymnosperms, wood consists of xylem tracheids. So it is softwood. Okay, so let's see the differences in between the cycas and pinus. Okay, so let's see the differences. First of all, let's take the wood. So in cycas, wood is monoxylic, and in pinus, wood is pycnoxylic. Wood is pycnoxylic. That means in cycas, wood is soft, and in pinus, wood is somewhat a little bit hard. Okay. In cycas, you can find only a few tracheids. That is the reason in cycas wood is soft. You can find only few tracheids. Only few tracheids are seen and plenty of cortex and plenty of cortex. And this cortex is made up of parenchymatous cells. Okay. So in pinus, you can find only a few cortical cells, and more tracheates. Okay. And in cycas, in cortex, starch is stored. And in pinus, there is no cortical cells. That means they have only few cortical cells. So here, no reserved material. Okay. In cycas, wood is not durable because it is soft. Not durable. Why? Because it is soft and it has more cortical cells. And in pinus, it has more tracheates. So it is durable. Okay. And in cycas, wood is not compact. Whereas in pinus, it is compact. Okay. And in cycas, wood has starch, which is stored in the cortical cells, and that starch which is stored is edible. It is called as sago. Okay. Next, coming to the pinus, it has no reserved material, and this pinus wood is used in construction. Okay. So the endosperm is haploid in gymnosperms and is formed before fertilization. Let's see. The endosperm. So these are the differences. Okay. Now next point. The endosperm is haploid. Endosperm is haploid in gymnosperms and it is formed before fertilization. Okay. Formed before fertilization. Whereas in angiosperms, endosperm is triploid and it is formed after fertilization. Okay. You have already seen the endosperm in angiosperms. It is triploid. Why? Because at the time of fertilization, one of the male gamete which fuses with the secondary nucleus. That means the polar nuclei. Okay. So this polar nuclei which has two nuclei and the male gamete which has a single nuclei together forms the triploid endosperm. So this is the reason why the angiosperms have triploid endosperm. Okay. So it is formed after the fertilization. But in gymnosperms, endosperm is haploid and it is formed before fertilization. There is a difference between the gymnosperms and angiosperms, between the endosperms. Okay. So this is the class about the gymnosperms, male, female gametes and the male, female cones or strobili, and also about the vascular bundles of gymnosperm. So this is the class about gymnosperms, whereas in next class we will discuss about the angiosperms.